In this mini lecture, we're going to talk about that first breeding scheme that I showed you in the last mini lecture. So here it is again. We take our existing plant or plants, usually it's more than one, we make a cross, and by sexual propagation we grow out those seeds and see if we have produced our desired plant. Once we successfully produce the desired plant, we will then asexually propagate it for distribution so that we have uniform daughter plants of our desired plant. So how do we do this? How do we make crosses? Well, for plant breeding, when you're trying to pollinate specific parents, what you do is you select a desired male parent, a desired female parent, and the idea is that one parent will have one characteristic that you like, the other parent will have another characteristic you like. You will cross them and hopefully get offspring that contain both of those desirable traits. And that's called a controlled cross. Now, controlled crosses can be done by hand, by insects, and then the last one there, wide crosses. This is not a method of making cross. It's a specific type of cross we make to achieve certain goals. Now we can do this to plants preferably in a greenhouse, because in a greenhouse there's no wind, there's less insects to interfere, because we don't want any pollination occurring other than between the two parents we've chosen. If you do a controlled cross outside, not in the greenhouse, such as in the picture here, what you're going to want to do is make your cross and then cover it up with a plastic bag or something like what's shown here to prevent it from being pollinated by anything else. We want to know that the seed produced from those flowers is just seed that resulted from the cross of the two parents that we intentionally put together. A few terms that it's important to know in regard to pollination and making controlled crosses. The first one is selfing. This is when we have pollination happening within the same clone. So this could be self-pollination within the same flower, or two different flowers on the same plant, or two different flowers on different plants that are genetically the same. So if they're two plants of the same cultivar, they should be genetically identical, and so if they pollinate each other, it's still considered a self. Now maybe you're thinking, why bother? If it's a self, are we getting anything new? Yes, we are. Because remember, it's still sexual propagation. So even though you may be doing pollination of a flower that is genetically identical to the flower that provided the pollen, the seeds produced from that will still be genetically different than the parent plant because it's still sexual propagation. Now, a cross or cross-pollination is when you're doing a pollination for two flowers of a different clone. So this would be one flower from one cultivar pollinating a flower from a different cultivar, or a different species, or a different genus. That's usually about as far apart in relatedness you can go with it. Still working in with genus, you're really kind of pushing it. So we have selves and crosses. Another term to know is dehiscence, and dehiscence just means splitting. We'll talk about it again when we talk about fruits and seeds in more detail next week, but in this case, we're talking about splitting of the anthers. When the anthers first form in the flowers, they haven't released their pollen yet. It's only when they dehisce or split open that the pollen is released. And so this is important when we think of our second term, emasculation, which is removal of the male parts from the flower. I know, it sounds a little gruesome, but that's what's happening in the picture there. They're using forceps to remove those anthers. And the reason you do that is this. Let's say that you do not want to do a self. You don't want that flower to pollinate itself. You want to do a cross. You want to cross it with a genetically different flower. Well, there's just one problem. We have a perfect flower here. It has both pistils and stamens, and so if we're not careful, it's going to pollinate itself whether we want it to or not. So what you have to do in this situation is to cut open the flower just before it would open naturally. So the petals should have the coloration that they're ultimately going to have, but the bud shouldn't have opened yet. You're going to slice open that bud with your forceps, and at this point the anthers will not have dehisced yet, so they won't have released any pollen yet. And so you'll slit that bud open, emasculate it, and then you can come in with the pollen you've collected off the parent you want and dust that onto the pistil. So that's how that process works. And now this picture on the left is a very old picture, but here is someone hand pollinating a plant with very small flowers. The picture on the right shows someone emasculating a lily. And lilies are really easy to do breeding on at home. In fact, a lot of the lily cultivars that are out there have been bred by amateurs. The nice thing about lilies are all those sexual organs, the pistils and the stamens, are really big, and so it's really easy to work with. So all you have to do for lilies, again, come in and emasculate your female parent. So pick off those stamens before they dehisce and release their pollen. And you're going to want to be careful with that, because if you have any experience with lilies at all, you know that those anthers will stain you. So don't get it on your clothes. It's going to take a while to wash it off your hands too. So you emasculate your female parent, then you're going to go collect anthers off of the male parent that you want to cross it with. And usually what we do is we put those in a dish. You can dab up the pollen with a paintbrush and then just paint the pollen onto the stigma of the pistil of your mother plant. Now you can see for this to work, you need to have flowers that are blooming at the same time. If they're not, you want your male to bloom first, so you can collect the pollen off it, put it in a dry, cold storage, so put it in your refrigerator until your female blooms, and you can take it out and use that pollen. So that's hand pollination. You're just doing it. You are doing the work of the bee yourself. Speaking of bees, our next technique is insect pollination. Sometimes if flowers are very small, it's almost impossible to do it by hand pollination. And if those plants are self-incompatible, that gives us the opportunity to use insects instead. Now they have to be self-incompatible, because if they're not, then they could be doing selfs. They could be crossing with themselves and we wouldn't know it, instead of having our two desired parents crossing. So for insect pollination, we're using this for plants with really small flowers that are hard to pollinate by hand, and plants that are self-incompatible, so we know they're not producing selfs. You could do it even if they weren't, but you just have to keep in mind that you'd have a lot of self crosses in your offspring and not just cross-pollinations or seeds resulting from cross-pollination. So on the left here, you can see how we do this. This is a cage in the greenhouse, just of sort of a mesh cloth. The two plants which are self-incompatible that we want to cross are inside that cage, and then they've released the pollinator insects inside. The way we usually do this, you can actually order the larvae of insects from a catalog, just like you can order anything from a catalog. They ship them to you, you put the larvae in the cage, and then once they reach their adult stage, they'll do your pollination. So the plants in this picture were pollinated by flies, so we put fly larvae in there, maggots, and then once they became flies, they did the pollination. Some plants, like the red bud shown in the picture on the right, are pollinated by bees, so you do the same thing just with bees. And actually, these red buds are the result of a controlled cross done by my advisor, again a plant breeder. He took a red bud with variegated leaves and crossed it with a red bud with purple leaves. And what he got were these offspring, and shown in the picture here, they have variegated green and white leaves, but the new leaves, the new foliage that come out this hot pink color, and then they sort of fade to a pinky purple color. So I'd buy that. I don't know if you guys would, but you can see you can get some really cool things by doing these controlled crosses. Finally, let's talk about wide crosses. The goal of wide crosses is typically sterility or infertility. So this is something we'd use on those invasive plants to try to get a non-invasive cultivar. And the reason this works, the easy way to explain this, is horses and donkeys. If you cross a horse with a donkey, what do you get? You get a mule. If you didn't know that, there's your animal science for today. Now, what if I want more mules? Well, I can't get more mules from mules. Mules are sterile. So if I want to get more mules, I have to cross more horses and more donkeys. Same thing with plants. Here are two plants I worked with for my research. On the left, we have the loblolly bay. On the right, we have the Franklin tree. I have their scientific names here. Now, neither of these are invasive. They're actually very nice, non-invasive native plants. However, they don't have anything more closely related to them than this. And this is the key. For a wide cross, we're taking two plants that are not very closely related to each other and crossing them, and that's what gives us our sterile offspring. That's why mules are sterile, because horses and donkeys are related. They're related enough that we can cross them, but they're not that closely related. And the same is true of the loblolly bay and the Franklin tree. They're not very closely related. They're in different genera, but they are both the only species in their genus, and so to get what we wanted, we had to find something to cross them with, so we crossed them with each other. And what we got was a plant that we called the Gordlinia. See how we mashed together the names. The Gordlinia was sterile, so we weren't really going for that, but you could see how you could use this if you were going for that. Take our burning bush. We'd find something that's just closely enough related to burning bush that they could cross and actually result in viable seed, but that aren't very closely related. And we could try crossing those and seeing if we got offspring that were then sterile. So that's a wide cross. We're using that to get sterile offspring by crossing two things that aren't very closely related to each other.